So this is one of the good beginning of the private-public partnership in India, I think. But we have a lot of things to be there. And what our request from the public sector is the actually allied background, genes in the allied backgrounds. So whatever available in present, I think IARI has done a lot of work on that. I think now they have started sharing with the MOVs and also on the payment basis. So that is one of the best things we should recognize. So if we do a lot of background work and clearing the backgrounds and coming with allied background material, that will be very good opportunity for the public sector, private sector to take away from the to the market. So, and apart from that, we look at a lot of, I think, Dr. Chandrasekhar has talked on the genomic selections. So, which is one of the really big thing happening in rice. And I think after the corn, I think the rice is one of the things which is most of the people are using, especially accumulating the favorable haplotypes in our backgrounds, our germplasm backgrounds. So that is really helping us to break the yield barriers. But still we have a lot of issues because we deal with a 3-line system, ABR system. So we need to have a lot of things to taken care of that. So, but commercial breeding, we look at only thing what is required for the farmers. So we address that. Maybe the minor one, actually hygiene is one of the things, even it's a market, people come back with a smaller issues also if he is not working in that geography. So if you want to take overall, it is major diseases, but I request public sectors to work on the minor as well as. So we should be future ready for the things. Thank you.
Thank you, Dr. Ramachandra. We have here listened to diverse views from our representatives from private sector. Now let's jump into public sector. I request Dr. S.K. Prashanthi, who's leading the biotechnology program here at US Dharwad as professor and head biotechnology. Madam, your views?
Yeah, thank you, sir. Thank you for this opportunity. First, I would like to highlight about the blast disease management. So our previous experience as well as the past experience witnessed the importance of PI54, PI9, and then PI2 interaction individually as well as in the combination. And I would like to say that the PI54 with PI2, it is found the best for our condition. So before this, the very important thing as an academician, I want to say that gene deployment or R gene introgression or the pyramiding should be based on avirulence gene information. So that should be done at the national program, like at the national level. So for that, there should be we need the helping hand from the national partners, and it should be a program in the universities or in the different academics. So the gene integration should be for the location specific, because Bagnaporte vora is highly diverse and a lot of transposable elements are there. It's all based on our student study, I am telling. So that's why the gene integration, fine, PI54 is, and PI9, PI2, in South India it is performing good, as well as in the North India also. But what is the future for us? So my second thought is that, why can't we just think about pyramiding the NBS LRR with the other type of resistant gene? So that is the second thought where the academicians have to work, and then the private people can take that information to transfer into their different varieties or the hybrids. So this is regarding how best we can manage the blast for the durable resistance. And because the R genes, there are plenty, and then majority of the R genes are characterized, but the avirulence genes, still we have to go further. So this is regarding the blast. And then the next thing is about the BLB, because in the upland situation, we never heard about the severity of the bacterial leaf blight. But since a couple of years, because of the rainfall, erratic rainfall pattern and then the climate change, we are experiencing the incidence of bacterial leaf blight in our situations, particularly in the drill zone situation. So we have to address it anyway. The genes are there. We have started pyramiding with the known blast genes, with the BLB genes. The program is there, already started in our university. And then the next challenge is the major challenge at the national level is the sheath blight disease management. So for that, we are stumbling at the first stage itself, that is identifying the different resistance source. For that, actually, I feel the work has to be still progressed where we can identify the very good source. Already the TETAP is known, which shows the moderate resistance, but there is any chance to find out the highly resistant source. We have the good germplasm collection, so we have started working, and then we have identified some of the highly resistant land races in our university and as well as in our germplasm pool. So again, it should be done at the national level so that we can go ahead with the management of sheath blight, which is very tricky. Already we have transferred the QTL, QSBR 11-1, with the PI2 and then PI54, which are all there in the testing resistance. It is showing fine, but at the same time, we have to select the lines for their yield and then the quality parameter. Here, the rice, the quality is also the prime importance. So these are the major challenges with us in front of us, and then for the sheath blight, still the long way is to go, and then coupled with the false smut. So we have to address these issues, and also again, several forum it is being discussed about the leaf blast and then the neck blast problem, and then how best these genes are the different genes. Can we have the such kind of genes, or the totally they are different types of genes that should be addressed? Even we as the academicians, we have to do some groundwork regarding that, and then fine. As my experience, 9 and then PI54, both at the both level, leaf as well as the neck blast, it shows resistance, because our center, that is Agriculture Station Mugad, is known for hotspot for leaf as well as the neck blast. So private people can take the advantage, and then we are there to work on that, and then you can share some of the lines if you particularly want to screen for the neck blast, where actually this has to be addressed on the priority. So this is all my viewpoints. Thank you.
Thank you very much. I now will be floating one question to each one of you. First, let's start with go in the reverse order. My first question is for Dr. Prashanthi. I'd like to know, are breeding programs particularly relevant to blast resistance, since you are also leading a program in this direction? Are they fast enough to stay ahead of evolving the pathogen, more virulent pathogen pathotypes or biotypes? What's your opinion?
Yeah, we are, because our choice is good, because they are the stable genes, whatever in the program we are at the national level, like we have seen 9, 2, and then 54. I think for a long period they can sustain, because we have seen the other part also, because AVR 54 also in the NIP program we have seen that part, so that's why it is hoping so. But anyway, we should not keep silent. We have to go ahead with searching for newer more alleles for that.
My next question is to Dr. Ramachandra. Sir, I would like to know whether we should continue. See, most of us have been deploying something called as marker-arrested gene pyramiding. We should continue doing this, or possibly in, say, another few years, we should go ahead with genomic selection in full steam. What's your opinion, particularly considering the pest and disease scenario in our country?
Yes. I think at present scenario, maybe another maybe until you get the germplasm loaded with the most of the traits, favorable traits. So we need to continue for maybe few years, maybe I see 2, 3 or 4, 5 years at the most. After that, I think the genomic selection is the only answer for all the pests and diseases, including yield. That's what my...
Thank you. My next question is for Dr. Chandrasekhar, who's the Rice India breeding lead from Cortiva. Sir, my question is, how many, you have transferred several genes in your breeding program in different crops. My question is, how many genes or QTLs should realistically be pyramided before agronomic performance suffers, or the balance between like a multiple gene deployment vis-a-vis agronomic penalty? What are your views on it?
Yeah, I think it also depends on like which disease, sir. Like for example, blast.
As a blast,
Yeah,
good example.
blast is like you will have breakdowns because these fungals generally they will have faster life cycle and they evolve fast. So you need to keep on doing that one. And for example, BLB, which is still very endemic to some areas, not very widespread, and also it depends on seasonal to seasonal. You need to be very, and it's like the most important factor which as a breeder I see is like if before deploying, we create base and converted and see is there any effect. So you need to see that for each of the gene. And for example, my experience, it may be different from others, but BLB have some drag is there, or some like blast, we don't see that much. And BPH, we have deployed a gene and in fact, we have seen reverse effect. Actually, it is positively influenced the performance. So it depends really on the question, on the gene in target, and also your own germplasm, because I cannot say what works for others. So you should know your germplasm and see before in that deployment, and based on that, you have to take a decision. There will not be a single standard answer for this.
Thank you, sir. You aptly put it. My question to Dr. Venkatesh Kulkarni is, what is the single biggest bottleneck slowing progress today in terms of large-scale deployment of molecular markers or genomic tools in breeding? From a private sector perspective.
From a private sector person, sir, if you are saying it is only in rice or in
any crop.
In other crops.
Why, like say, the progress with regard to molecular breeding is good, but it's not excellent or outstanding?
Yes, yeah. I go by you on that front, that it is very good, but breeder-ready markers in most of the crops are not available. For example, if you ask me as a private sector, after this rice bacterial blight and blast, and BPH markers, in which crop you need breeder-ready markers? That I say that pearl millet blast. Pearl millet blast is one. We can take the similar line and develop. It is one of the most deadliest. Now, downy mildew is not a big problem in pearl millet. It is blast now. So breeder-ready markers, in maize at least, we don't have one. In millet, we don't have one. So in cotton, we don't have one. So we really need a breeder-ready markers. Those can be deployed very well. And of course, backcross method, which is to be used for integration purpose, a lot of us, we have standardized fastest backcross protocols, wherein background and foreground selection can be done, and we can deploy them as fast as possible. Coupled with now with speed breeding facilities, this can be done very, very quickly. Like speed breeding facilities and double haploid facilities of CIMMYT in maize, we can do very fast. I presume that with the success that what we have seen in rice and even tomatoes, the lot of efforts on developing breeder-ready markers for different problems that we have associated. Another experience with me is that if you ask me markers for insect tolerance and markers for disease tolerance, I prefer markers for disease tolerance would be more successful than insect tolerance with the experience that we have. So in such a case, the private sector, which has got good investments in developing hotspot, probably most of the private sector is also into heavily on line development programs also. That means to say backcross breeding, forward breeding are integral part of our breeding. So in such a manner, when all these are available, only thing, if you ask me that, okay, can we have breeder-ready markers developed from the private sector? When there is a question mark on the result of the experiments, in private sector investment gets curtailed. So we are seeking some proposals through BIRAC or DBT to get the grants and develop for important. In our company, we have listed 10 biotic stresses as important, and we are working on them on all fronts, including transgenic gene editing, conventional breeding, and management approaches also. So I love the marker-assisted selection, and we need some weapons and tools to handle them in different crops. Thank you, sir. Thank you.
My next question is to Dr. Devaraj Achar from Indo-American Hybrid Seeds. My opinion is that we are over-depending on major resistant genes, particularly in rice. My experience is limited to rice, and we should focus more on QTL-based resistance also. So in your opinion, based on your experience in multiple crops, what should be the ideal balance between major R genes and QTL-based resistance?
Yeah,
Take
exactly.
any one crop as an example, sir.
Yeah, if you take any crop, of course, you have a monogenic, oligogenic, and polygenic. If you see the same example for the blast, earlier we were talking about 1 or 2, 3 genes. Now the concept of monogenic and oligogenics has gone out now, because the multiple genes are being count. If you see the particular gene, the phenotypic variance of that particular gene, hardly it gives 30%, 40%, something like that. So we call it as a major genes. Now we call it as a major QTLs. The QTL approach is the best approach to introgress to any background, so where we can able to bring a maximum resistance. Even in the QTLs also, you could see that major QTL again minor QTL. So this is a very complex. So it depends on the what kind of a trait which we are targeting. For example, if you talk about the BLB in rice, hardly, as you said, these 4 or 5 genes are enough. But if you talk about the blast, it's not so. We are claiming PA54, PA2, PA9 gene. But if you take the validation, the level of validation, if you take the publication versus validation, we have more publication than the validations. So the lack of validation, sometimes it gives the inconsistency whether I should use the markers or not. So there is no proof of concept. See, for example, if you take the buyers which have been released, the rice, so hardly they have 4 or 5 genes and PA2. So if you see any private companies, every successive years or every 3 years or 4 years, absolutely they have to release some new hybrids, otherwise it is not possible. So the complexity of the diseases keep on increasing. So plants cannot combat immediately after introgressing 1 or 2 genes. So therefore, I would suggest, it's my feeling, so my experience saying that complexity of the diseases is more. So therefore, QTL approach is the best approach where you can combat the multiple races, in fact. So not a single gene which is particular race. So that would be the best approach. And also as well as the genetic background. Sometimes if you see a particular variety, or if you have 10 varieties, you are introgressing multiple genes or multiple QTLs. But all 10 varieties which have been introgressed, they will never perform the same way for the resistance sometimes. So it is not a monogenic or polygenic, but it is oligogenic as well. But the genetic background which always complement your whatever the targeted QTLs. So we need to think about those genetic background as well, rather than focusing on those targeted QTLs or genes. So QTL approach is the best approach.
Thank you very much, sir. Now I have a question for Dr. Viresh Koda. Bayer, I understand, has invested a lot in multi-stress resistant or tolerant breeding, not only in rice, but also in many crops. What is your outlook in terms of multi-stress resistance breeding? You can take rice as an example. Say in 2035, how do you look at the scenario?
Yeah.
What actually we'll be doing?
Yeah, sure. I think I'll go back to one of the suggestions made by Dr. T.R. Sharma, sir. Sequencing is very cheaper now. We can make use of that sequencer. How can we generate more insights, and also to address some of the the challenges what we have with new pathotypes which keeps emerging with the cloud of climate changes. How can we go and start using those information and bringing that part of our genomic selection, which takes care of all the required traits for the customer. So I can say maybe we have gone through some of the era on the marker-assisted breeding or MEBSA. I think the way forward approach should be to go with the sequence information, which is much, much cheaper. I think even $100 what Doctor was mentioning, it is even lesser if you start thinking of a small crop like a rice. It can be done even at $30 per sample. So how can we get into that to tackle those is the approach which we have started doing already, and I think that's the way forward we are looking at, particularly in the Bayer.
Thank you. We had perspectives shared by representatives from both public and private sector. Before we close today's meeting, I would like to invite one or two students to ask any questions which are pertinent to the topic of today's discussion. Any questions from the students? Or even members of audience can also ask pointed questions to one or more of our panelists. Okay.
Thank you, sir. Since the whole lecture, in the morning, it was very interesting and people were talking about rice. It is not directly on the topic, but my concern is about instead of developing disease-resistant variety for multiple traits, can we focus on the indigenous variety which is very popular in a local area like Kala Namak or brown rice or red rice? What are the efforts being done for increasing the yield of those varieties which are still localized in certain area of India? Thank you, sir.
That's a question I can answer because with regard to private sector, I think Prashanti madam can also answer. Private sector, the investment with regard to local land races may not be there. Madam, maybe do you have any perspective to share?
Yeah, we have witnessed the very good performance of the land races. They are unique for maybe the quality or for the resistant genes. That also we have already using those land races, resistant land races, and then identifying the genes and then transferring from that.
But the yield is the constraint in those varieties.
Yeah, but we have to take the permission from the biodiversity board, and also as I know there are some issues regarding we cannot improve the land races, but we can do the selection and then conventional breeding we can practice for the improvement of the land races. Anybody can add to this?
Well said by Dr. Prashanti. I would like to add one more element to it. One of the most easiest ways in which we can manipulate or improve land races or land cultures is through genome editing. My colleague Dr. Satyendra has already started doing this in one of the local land races, of course with appropriate permissions from the community elders. He's improving a local land race called Chitti Mutyalu, which is known for its aroma, small grain size, and then extraordinarily high zinc content. He's working to improve because it's very tall, it has got photosensitivity, and it has got a few other problems. So by targeted editing of a few genes, without changing the genetic background much, so that the useful traits will not be lost. So genome editing could be one among the many strategies which we can deploy for improving local land races. That's my sir.
I want to have one perspective because Nath Biogen has one variety called Nath Poha, which is very, very popular. We sell a large quantity of that. It's fantastic poha making ability of that. We employed an MBA intern for assessing that. By inspiring that, we have a small breeding program. We spend around 10 to 15% of the time called Specialty Rice Program, which is called Use For. So one of our board member brought up an issue saying that why should not be poha be in pink or red color? So we are now currently introducing semi-dwarf gene in those land races which are normally tall and bold seeded. So we have some red rice developed in semi-dwarf. Only thing because our source of semi-dwarf is not very bold. So 2-3 more back crosses have to be done for getting semi-dwarf and heavily bold. The seed weight is between 33 to 36. So this is one, only we are focusing on semi-dwarf. On biotic stresses and other kind of things, they should follow. I know sources are very limited and we should do it in the future.
Thank you.
Thank you, sir.
Any more questions from any members of audience, please? Can I ask a question in general or ask a relevant panelist also to answer? Okay. I think.
Hello.
Yeah.
Good afternoon, sir. I am.
Yeah, go ahead, sir.
Yeah. So I am Chidanand.
Second last question, okay?
Yeah. So from Kote of Agri Science. So my question is, we have seen one virus, like rice DORP streaked virus in Punjab and Haryana area. So I have not seen anything in this public research. Now, you have not yet started, I think so. So my question regarding that, we have any tolerance genetics available for this disease, or it may not be a threat to the rice production in India?
My perception is that the disease comes sporadically. Just like private sector, we need to prioritize our investments. So what we have decided as part of All India Coordinated Research Project on rice, we have identified 2 research stations. One, our main rice breeding station at Ludhiana as part of PAU's group. And another ICRISAT station is located at Kaul in Haryana. Around Kaul locality also, we observed a lot of incidents even this year. So those institutes are working. And PA has developed a field-based screening protocol through ELISA, wherein quickly you can identify the infected plants. And Kaul together with PA is working on identification or validation of the resistant sources. But this is a local problem, particularly in northwestern part of the country. The entire country cannot work on it. So we have prioritized this particular problem for Haryana Agriculture University Rice Research Station located at Kaul, and Punjab Agriculture University, their station at Ludhiana, to focus on this problem. Thank you.
Thank you, sir.
Last question, please.
Pathogen and environment. We tap anything like a host. In the host, we do everything we need from that side. So in pathogen point of view, how far we came to know the pathogen biology, and whether, say for example, if we collect the isolates from different hotspots, we screen for most virulent isolate, and we check it in some other place. So we got some resistance in that location. If we take that variety or a hybrid to particular hotspot, it becomes susceptible. So my point of view, how far we know the pathogen biology relevant to particular disease. And my second question is, we talk about the terms like resistance and tolerance. When we have to use that? See, for my view, tolerance, in case of tolerance, if the pathogen and host is fighting, and the host is...
Sir, since time is short, I got your 2 questions. First is regarding...
First question I will answer. So in the beginning only I told that we should once, before we start the breeding program, we should have the perfect knowledge about the pathotypes or the pathogen variability which is there, or where actually our product will go and then hit the market. That information, based on that only we have to start, and then we have to choose the origins.
Yeah, there are multiple ways we can learn and know, but one simple way is always, we do conduct multiple multi-location trials. How to enhance the same opportunities to monitor the pathotypes or even whatever the races we want to know. If we can collect and start monitoring over the years, we'll know a lot of diversity and also the trend, the change, whatever is happening. Maybe that is one approach. I think it can be helpful as a simple way.
So adding to Veera Gowda's this one answer, you cannot forecast how quickly the pathogen is going to be changed, in fact, okay? So only the way is every season, every time. So we have to collect, and we need to, whatever the pyramided lines which we have, keep on screening only. That is the only way where we can able to say that, yes, whether I can go for this year, next year. The moment what we have been developed for the variety which is resistant for the same hotspot, so another next season itself we cannot expect that particular hybrid is resistant. So therefore it is a continuous process. We don't know exactly in what way they are going to mutate and how fast they are going to mutate. So it is a continuous process as a pathologist. So we should coordinate with the breeder who should work together. Then only we can able to come out something. Thank you.
I think we cannot take any more questions because of paucity of time.
So just want to add about his last question about resistance and tolerance. As a biology student, we should never use resistance, because we just talked about like next season only it may become, so there is nothing called in resistance in the biological world. There is a lot of multiple things, environment, host interaction, how it is evolving, races. So you should only use tolerance. That's my perspective.
No. It should not be like that.
Because viruses, we don't want to use the word tolerance.
In viruses, sir, I'm telling about more
when you
about
said biology,
pathology.
that's why I said, in viruses, we don't want to use the word tolerance because in spite of the yield, there will be source inoculum will be there for further spread. So never select a tolerant variety if it is a virus. My other point, which I thought time is over, but I'll still share. In many horticulture crops, resistance is breaking down at high temperatures and extreme weather. My suggestion to all the panelists is also subject your genotypes to slightly extreme weather and see their performance.
That is something very well taken, sir. So now I'd like to close today's discussions. We had sharing of viewpoints or perspectives by many members from private sector, and we had one lone but not the least member. We had one representative from public sector, Dr. Prashanti, who shared the perspectives, particularly based on her experience regarding management of blast disease. And I can take at least 4 or 5 concluding points or points which you can take it from today's panel discussion. One, I would like to mention that resistance breeding program must also be dynamic, just like the way the pathogen and pests are dynamic, and they should be multi-layered. And along with deployment of the major genes, we should also explore increasingly the QTLs or the minor genes also. And I would certainly say that future breeding will or certainly should involve deployment of large-scale genomic tools, artificial intelligence, and also speed breeding. And we should not take away the fact that continuous monitoring of pest or pathogen evolution is the need of the hour. And along with resistant gene deployment, we should also do how these genes work at different locations and how the pathogen is evolving to overcome the different resistant genes. Last but not the least, collaboration across breeders, pathologists, entomologists, and data scientists, along with molecular biologists, is vital for success. I'm talking from my own example at IARR. We had a cross-section of team of molecular biologists, pathologists, entomologists, and also other trait specialists who worked together to deliver a product in terms of gene identification or gene deployment. And ultimately, farmer adoption is the ultimate success metric. And I'm sure that in rice, at least, we have recorded a lot of success stories wherein varieties with single trait resistance or multi-trait resistance or hybrids, they are increasingly adopted by the farmers. So I'd like to thank each one of our panel members. Yes, sir, just quickly.
No, sir. I just wanted to say because today rice breeding sets an example of in two-way direction of crop improvement. One, they are attempting to reduce the losses due to biotic stresses, and also they are increasing the yield by manipulating yield genes. I have visited his program, and both way, it was a visual delight when I saw the yield gene plot. So I think other programs can also be on this, increase the yield potential of the varieties, plus also reduce the losses due to biotic stresses. This two-way approach will enhance significant productivity in each of the crop, and I salute the IIRR's efforts in this, and the way in which the field visit was something which is memorable to my life, sir. Thank you for that.
Thanks, Dr. Venkatesh Kulkarni. I would like to thank all my panelist members for today's panel discussion and also the audience for active participation. Thank you, one and all.
Thank you, sir. Before we move for lunch, we take the honor of felicitating our fellow panel members. I request chair of the panel, Dr. R.M. Sundaram sir, and also Dr. Viresh Gowda to felicitate other fellow panelists. And I request all the registered delegates to carry the food coupons while you go for lunch. And there is a special announcement to students. There is a special interaction session with the industry personnel at 6 o'clock. The interaction between students and the industry. And from today evening, all the registered delegates can collect their registration fees receipts at the registration counter. Now we'll break for the lunch and we'll move to the guest house to have a lunch, and vehicles are waiting outside, and we'll join back again for the next session at 2:40. We'll have a lunch break of half an hour, and then we'll assemble at 2:40 for the next session on Maize. Thank you.